So thank you very much to UNC for extending its arms to us today and allowing us to take some of your time as well. So the title of my presentation today is Metabolomics Advantage, Elucidating Gene Function, Causality, and Therapeutic Relevance in Population Health. So we all are very comfortable with the hierarchy of biology, right? We have a genetic code that translates into proteins. Proteins conduct a function generally by, creating changes in small molecules, right? So we really think of metabolomics or small molecules as the closest to the actionable phenotype. If you have actual changes in metabolite levels, ADP going up or down, glycine going up or down, glucose going up or down, you know that everything in the hierarchy up to that point has had to be shifted. Could be altered protein transcription, could be altered post-translational modifications, alternate splice variants, all the way up to different mutations in the genetic code that are leading to that ultimate signature. So metabolomics is often used in population health to bring greater knowledge to what we're seeing in the genetics, right? Offering that Rosetta Stone of unlocking what are the penetrant mutations? What are those variants that are of unknown significance? There's lots of genes we don't know the functions of yet, right? Looking to see what metabolites they correlate gives you that snapshot of what is this gene actually coding for and how is that playing out in biology. But, and this is always my but folks, there's more slide, right? Not only does it give us that view of your entire endogenous hierarchy, it also captures the complexity of being a human living in an environment, right? We all know the huge impact that the microbiome has on our health. It is not just our own genetic code that dictates our health. It's our environment. It's the state of our microbiome. It's the drugs that we're taking, the molecules, the pesticides that we're exposed to. All of those things play into complicating our analysis when we study humans, but metabolomics is one of the few omics that actually allows you to start teasing those factors apart. I often think of the example, back when statins were first introduced to the market, if we had run a metabolomic study, we probably would have seen the grape juice metabolites associated with higher metabolomic or with people that had that sort of aberrant factor with grape juice showing elevated levels of the statin, right? So if we had metabolomics back then, that might have allowed us to really comprehend that more complex dynamic between host and environment. So one of the most powerful approaches we've seen is in the study of population health. And why do we study populations, and why is it so important to study these in big numbers? And again, this is not anything that most scientists already are very comfortable with, but because humans are so variable, you have to power through those differences. We have different diets, we have vastly different genetic backgrounds, and histories, as well as health statuses. This is a great example just showing that if you studied 5,000 people, you can't actually clearly see the trend that we know that as you age, your blood pressure tends to go up. But once you study 500,000 people, you can absolutely see that. So it's that greater numbers that allows you to tease apart some of the more complex phenotypes and really start capturing those complexities. Metabolon really first got interested in, really this sort of ability of metabolomics to feedback and inform on genetic information over a decade ago. And some of this really early work, this was actually some of this was work which published back in 2011, was done where we ran the Twins UK cohort as well as a Cora cohort out of the EU. And what they were looking at is, again, looking at association, gene mutations with specific relevant metabolites and finding where those connections lie, right? So not only did we see incredible information about how tightly mutations in certain loci directly correlate with specific metabolites, they were so tight that you could actually see heterozygous versus homozygous. If somebody was a carrier, you could see the metabolite levels titrating, and you can see the example here for two particular mutations. Let me get my mouse up over here. Two particular mutations. That you could see that the person across these two completely separate patient population cohorts, that the metabolite levels correlated and titrated with carrier status as well. So that was really surprising to us. It's not just that they correlate with extreme precision, so that not only can you start to understand which mutations are truly penetrant, you can also see, if you have a mutation that's a heterozygous, you can start to also classify, an example I'll show you in a minute, what it looks like. You know, in larger populations, you can start to capture more rare mutations, right? And again, this gives you more power. So this was a study, and I'm just going to be here briefly because this is my kickoff to the next slide. In the same population, they started to look at rare mutations and the metabolites they correlated with, and they found a really fascinating example of 3 different types of mutations found in this one DMG, dimethylglycine dehydrogenase locus. And the interesting thing was not only could you see, these were twin populations, so a nice internal control. The numbers that you are seeing here, sorry, let me get my mouse up. The 2 numbers you can see here are actually the 2 sets of twins, and you can see that these twins were monitored over 3 visits. This is actually 3 decades worth of monitoring these twins. So this is decade 1, decade 2, decade 3. And if you look at the numbers, I know it's a little bit small, but what you can see is that sets of twins had higher levels of DMG over 30 years. Another set of groups of twins had sort of a moderate, and then the third set of twins had a lower. All of these correlated with specific mutations. So not only can you see a mutation as penetrant, you can see the severity of the mutation, such that in this particular example, the stop-loss mutation had the most profound impact on enzymatic function in that particular mutation. The next was this missense mutation, and the final, still affecting their normal levels of dimethylglycine, but not as severely, was this stop-gained mutation. So again, it just gives you incredible power to understand the severity of the mutation, whether that mutation is actually doing anything. And you can see in this example, this tight correlation between gene function and gene metabolite. It makes perfect sense that you're seeing alterations in dimethylglycine for a mutation in dimethylglycine dehydrogenase, right? And we see those tight connections across the board. And this same research has allowed us to identify gene functions that were previously not known, and vice versa, where we find molecules that are unknown, right? We don't know what it is. We see it in biological samples. We don't know what it is. It's a signature on a mass spec. The gene says it is highly correlated with an R-carnitine transporter. Okay, let me see if this unknown is some sort of transporter. So the data goes both directions, in both informing what are the metabolites that we're seeing, as well as the back. The metabolites that we're seeing correlated to these mutations help us then understand what the mutations are. So it's a very powerful technology. Again, we've been running genetics for decades. We have scads of data on genetics, and we still don't know a lot. We don't know how to assess some of it. And metabolomics really helps. The studying of the small molecules really helps build the strength of your genetic data. Now, one of the things I find fascinating, this is actually just published last year in Nature Communications. The complexity we know about our genetic code is that any one molecule is not associated with one gene. For this particular molecule, N-acetylkynurenine, to exist, it has to have had the action of 5 different proteins, which means 5 different mutations in 5 different areas of your chromosomes had to be doing their job to ultimately get to this end product, right? So it's not as simple as one gene mutation or one. It is the combination of many that have to come together to ultimately influence this metabolite. And if this is a key metabolite in controlling a particular pathway, you actually have to understand that it's not just one gene, that there are multiple ones in play bringing this together, but you can see this all beautifully. On the flip side of that, you can see that there are also proteins that are quite promiscuous, right? They like to acetylate anything they can get their hands on, right? NAT8 freely acetylates a number of different amino acids. So in these cases, you can see gene mutations that actually affect a broad category of molecules. So that one mutation in that gene may ultimately lead to pretty widespread metabolic issues. Same thing with the aminoacylase, that is the deacetylation product. So both sides of this equation play into ultimately controlling the level of these various acetylation products. But again, this allows you to know that that mutation is penetrant. That one ultimately affects the ability of your body to control acetylation levels in your body versus other mutations that have no impact. Now, I am a big fan of Manhattan plots, but I don't know how people who are in genetics look at this stuff, because I look at these and it just blows my mind, right? But I think there's some incredible power, obviously, in people who are trained to like understand Manhattan plots. I'm always just like, you know, I bow to you because they're complicated, right? They show us how complex your genetics and the mutations at play can get when you're trying to understand phenotype. So this was a great work done by, again, one of our collaborators, published in Nature Medicine last year, where they looked at a population. And one of the things they were able to tease out from this population is they had a group of people that were being treated for male pattern baldness with a class of anti-androgen drugs, right? And there had been known or observed clinical ramifications that some individuals who are treated with these anti-androgen drugs have a susceptibility to depression and or suicidal tendencies, right? So there had never been a real understanding of what it was about these anti-androgen drugs, and there was no evidence about how these two were collected other than clinically they saw this happening. So in this particular cohort, they were able to look at both the genes as well as the metabolites that were associated with these two conditions, both depression and male pattern baldness treatment. And you could see from the two genetic analyses, while there was some shared mutation, they were mostly pretty independent signatures. There wasn't a lot of mutations consistent in male pattern baldness and consistent in depression to really show, here's the lynch, here's why these two things are linked. But once you add the metabolomics data, you can start to see that across the metabolomics, you see similar classes of molecules that are showing up that are actually now statistically significant to both. This was really the first indication bringing together these two somewhat disparate concepts of male pattern baldness and depression, and finally creating that link, because you can see that the play is in the molecules that are being produced in the treatment of male pattern baldness, in addition to those that are being created during depression profiles. So really allowing you to finally tease apart, why are these two things linked? We know they're somehow linked clinically speaking from what we've seen in patients, but now we actually have some evidence. Again, more insane Manhattan plots, but I love these. I read this paper and I brought it to Micaiah, who was helping me with my slide deck today. And I said, I see these. These are so amazing. We've got to put it in the presentation for next week, because they are just sort of breathtaking when you see the clarity of the signatures between mutations that are observed in these different locus and how tightly they match metabolic profiles. A lot of current gene work is about understanding causality, right? Like, yes, you can see that there's correlations between a mutation here and blood pressure, and here, or depression, or here and this. But the concept, what is the causal gene? What is the mutation that is actually related to the cause? And so a lot of statisticians are working today about finding that causal link. And often what they are doing is looking for these changes in not only mutations, but ones that correlate to known molecule changes in the body to really bring about that causality understanding. So this was from that same Nature Medicine publication from last year, looking at 3 different locuses and how they correlated with various phenotypic etiologies. So blood pressure was one. So, we had that there was particular mutations in the BDH locus that was tightly correlated with VMA, a metabolite present that again showed extreme consistency in how the metabolites correlated with each other and how they correlated with the mutation. Same thing with pulse rate. So pulse rate, there were mutations, and you can even see in the Manhattan plot that there's a series of different mutations all in the same area. And look how tightly that correlates with both the N-methoxytyrosine metabolite as well as dopamine sulfate metabolite. It's that one-to-one titration of, you see a mutation here that has some impact on the metabolite level. So you're actually stratifying the severity of the mutations in that locus and being able to directly correlate back that has impact on these metabolites. You can now start to look at how are those metabolites playing in blood pressure? What are the targets related? Why are those metabolites having this influence with with pulse rate? And then the last example here was breast cancer. Right, again, known mutation signature in the, I should use my mouse, in breast cancer in the PPM1K locus. And again, incredible correlation with several branched chain amino acid. We already know from literature that branched chain amino acids are absolutely involved in breast cancer. But again, this is just more evidentiary, really showing that connection between genetic mutations, genetic susceptibility, and seeing that actually execute in the metabolites that are being produced. So that was my whirlwind. I'm now going to focus, and instead of just showing you a cool study after cool study, I want to share with you one story that is maybe a little bit more grounded in scientific content, right? So this is really interesting work that we've done with Karsten Suhr, who is a big population health advocate and loves to run as many patient samples as he can get his hands on, because he loves being able to look for these associations. So as he was looking at this, he had a large cohort, and this was actually a fantastic study, because not only were we able to look at things like acetaminophen usage, we were also able to assess things like how well did people report their drug usage in studies, right? One of the fascinating things about any of the work that we do in this area is that we are all susceptible to what people report about what they've taken. Have I taken antibiotics? Have I taken Tylenol? Have I taken, ibuprofen? And unfortunately, most people don't know the drugs that they take or don't understand that. So if you say, in a survey or a question for a person participating in a study, have you taken Tylenol? They may have taken NyQuil. And so they say, no, I haven't taken any, none here. But you see all of the metabolites of acetaminophen in their plasma. So you know that they have consumed acetaminophen. They just didn't know that. And a really interesting from that study, and I'm off topic, and the guy's like, what are you talking about? That's not in the slide deck, Annie. I like telling these little side stories. On that same study was really fascinating findings about reporting the use of anti-anxiety medication. This happened to be a population. We had two separate cohorts, one from the UK and one from the Middle East. What we saw was rampant underreporting of anti-anxiety medications, with far more prevalence of underreporting in the Middle East than the UK. That probably speaks to the bias of the societal pressures about there being shame associated with anti-anxiety medications, more so in the Middle East, a little bit more accepted in the UK. But again, we could see the metabolic profile. We know the drugs were present in these individuals, even if they didn't report that they were taking it. So a fascinating aspect of, again, metabolomics and study population is that you can start to tease apart some of these self-reporting characteristics in individuals, because the metabolomics, the molecules present in the blood doesn't lie. It is a really grounding area of truth. So back on topic. So this was a study looking at acetaminophen. Acetaminophen has been studied, you would think, to the point that there is no new discoveries to be had in how acetaminophen is metabolized. We know that there are populations of people that cannot process Tylenol well. They actually accumulate very to liver toxic metabolites in their system, which causes extensive liver damage. And since this is a college town, this is why they tell you never to drink alcohol and consume Tylenol, because alcohol metabolism actually induces some of these metabolites that are known to be very liver toxic. So that's why if you're going to be drinking, take ibuprofen. I'm not an MD, don't listen to what I'm saying, but there is that. That's why that's their connection, is because alcohol does induce some of these toxic metabolites forming. But again, Tylenol, incredibly prevalent usage, #1 painkiller on the globe, right? And we are still learning things about how it is metabolized in the body from some of these new population work. I'm going to share just two different case studies related to Tylenol metabolism. Again, the mutations that lead to the inability to successfully catabolize or metabolize acetaminophen into less toxic and or just to clear it from the body is pretty well known. One of the things that we did is we had this, kind of a precision medicine aspect of this. We always sort of curious, like a lot of science that we do are discovery based, right? We take a big population of treatment and a big population of controls. We compare the two, we find out what's statistically significant. Oftentimes in population health, particularly for rarer disorders, you only have one or two, right? Or you have 3 or 4 or 5 or 6, right? You have a smaller number. So part of the analysis of this data is understanding how much power can be derived in fewer cases if you have that consistency of biological sense in terms of the genetic makeup and the mutations where they exist. So this was a study where it ran healthy self-reported individuals. And I love this because this is, I bet if you queried the room and said, are you generally a healthy person? Most of us will say, yeah, for me, I got a high BMI, that's not great, but generally a healthy person, right? So these were self-reported, 80 self-reported healthy folks came in, had their metabolomic profile, and there were 2 people that just had these I mean, astronomically high levels of acetaminophen metabolites present in their blood, way higher than if they had just popped 2 Tylenol before giving blood that day, right? Just really, really high levels. And that really made us suspect, are these people that potentially have a metabolic profile that doesn't allow them to process acetaminophen, therefore accumulating these toxic metabolites? So our next question within the very same data set was, are we seeing perturbations of liver metabolism? So looking at bile acids, a good indication of how well the liver is functioning. And the one patient that we were particularly concerned about that just had absolutely pegged, this is 300 times sort of amount of acetaminophen that you would see again in a group of people. Massive, massive signatures for these metabolites. But fortunately for this individual, we didn't see any consistent signs of liver malfunction or liver disease. But then we also saw that glutathione, this person happened to have very large amounts of glutathione. So they likely had a metabolic system that was sort of protective of the fact that they probably couldn't process Tylenol because they had all this glutathione, which prevented the liver damage. The other patient on the right, though, had a subtly different story. So while their metabolites were not as extreme, they definitely had signs of rocking on the higher level of liver dysfunction, right? So they were generally above normal in the level of bile acids. And if you, again, look at that protection of glutathione, this person had none of that glutathione to protect the liver. So this is why it's, again, these 2 individuals, self-reported healthy, probably had, now we didn't have the genetics when we did this initial precision medicine study, but probably had mutations that made them unable to really process Tylenol. And again, this was just 80 self-reported healthy folks. This is you and me. These are prevalent. We then did follow this up again with Karsten Suhr, who wanted to look at this in more depth. With this new data analysis, found several new mutations, SNPs that were associated with the inability to actually process Tylenol successfully. So 3 SNPs total under this UGB2B15. Again, that's already known to play a role, but then also found a strong association with this catechol O-methyltransferase, again, mutations in this gene. Again, this was sort of new and had not been really noted before. So again, even in something as prevalent as Tylenol, as well studied as as Tylenol, there are still new discoveries to understand the influence of genetic mutations and how they impact our ability to process something as common as Tylenol. I'm just going to skip that slide. The last case study I want to share with you, and I forgot to start my timer, so you're going to have to scream at me when I go on, was this looking at a gut microbiome. Now, one of the really interesting things, and one of the places we've seen the most power in that connection between genomic and metabolomics, is in the microbiome. One of the reasons for that is so many people right now are studying what microbes are present, right? They're doing 16S, they're identifying the specific phyla that are present in the feces or wherever they're looking. This was a really interesting study where they characterized a number of patients and did swabs and or samples from various parts of the body so that you can absolutely see the strong distribution of different phyla present across individuals, even when you do a stool sample or a tongue sample or whatever, wherever it is that you were testing. But if you look at the metabolic signatures of each of those different swabs, even with those dramatic different distributions of bacterial populations, the metabolic profiles are pretty conserved. So you don't necessarily, the power is not in what is there, but what is the metabolic profile of what is there? What are those bacteria doing? What are they producing? And what of those bacteria are producing the bad actors that are actually shifting the metabolic profile enough to cause, poor indications or outcomes. But so many people are still really thinking about, and most of the publications you're seeing are what phyla are there, what phyla is associated. But the next thing they're going to have to start thinking about is, yes, those are different, but those are essentially making up the same metabolites that are going to be present in the feces. What you need to start looking at, what are the bad actors? What are the ones that are producing aberrant metabolic profiles? And again, along those lines, early on, this was actually a publication. It feels like it was decades ago, but it was only about 5 to 7 years ago. Back when we were, still sort of really trying to dig apart the influence of the microbiome on health. This was a particular study where they had host whole genome sequencing. They had the RNA sequencing in the feces itself, as well as a bunch of clinical measurements and metabolomics, and wanted to understand the interplay between the host genetic code, the bacteria present in the feces, and which had more power in dictating the microbes that are present in the metabolic distribution in the feces. And perhaps surprising to absolutely no one, the strongest contribution to metabolic profiles of the feces, or what is happening in the gut, is the microbes. It is not the host that is dictating what is happening in your gut. It is the microbes in your gut that are dictating what is going on in your gut, right? And so while, of course, important to tease that apart and understand that there are influences between what is happening with our own genetic code as the host, but that the far majority of activity in your gut is being dictated by the bacteria that are present there. And then perhaps my favorite slide from, or my favorite figure from that publication was this one. We talked about how similar the metabolic action of a lot of these different bacteria phyla are. And this is just a visual way of showing that, yes, you can see that there are classes of metabolites. So across the bottom here are just different, different classes, xenobiotics and peptides and energy metabolites. And you can see, as you just let your eyes visualize this, that there are some molecules or some phyla, bacteria that, yeah, they produce that same class of molecules every time consistently, doesn't matter what phyla it is. But then if you let your eyes dance around, it's like, oh, but I see that there are some molecules in here, and those are only produced by bacteria. Oh, I can't even say it, by that, right? Or there are other molecules that are only produced by that. That is the power of the metabolomics, is really being able to tease apart what is the metabolic properties, what are the metabolites being produced by those bacteria that are present. So I'm going to wrap up and start to let Annie Green come to the stage, but I just want to, again, I want to bring up one other case study. There are applications of this even outside of population health. This was a wonderful study that I think I love. Anything in children just moves me to tears. This was looking at patients that relapsed from leukemia, right? So there was a specific population of children who would relapse, and 20% of time they had this specific genetic mutation. So they really wanted to understand what was happening, that this genetic mutation was leading to a higher relapse rate in these leukemia patients. And what they were able to find through the combination of, again, the genetic mutation and what we were seeing in the metabolomics is that the cancer cells the mutation was a gain in function mutation. So it had a better job of shifting that gut, or the drug, to kill the cell back out of the cell. So while the drug was getting in, that gain of function shipped that drug right back out again. And so it saved the cell, the cancer cell, from the drug because it shipped it out. The interesting conclusion of this, though, is not only did this gain of function ship the drug out of the cell, thereby making the cancer cell survive, it also shuttled other things out. And so it opened up other potential avenues to actually use the knowledge about what that cancer, other things that cancer is shuttling out, in terms of a way to try to find a way to kill those resistant cells. So again, this sort of, not everything are these massive population health studies really teasing apart complex phenotypes. Sometimes it's as simple as this single mutation causes relapse in this single type of cancer, and what we can learn by studying just that one relationship between the metabolites and understanding how is it that these drugs are escaping the cytotoxic effects of this particular chemotherapy. So I hope I have shared some of my enthusiasm. I know I'm a bit crazy. Self-admittedly, I'm a bit insane. I am very passionate about this work. I'm super excited. I consider it a complete honor to be working in this space, because I see that the work that I do every day is having a strong impact in shifting science, shifting how we treat people, shifting our understanding of human biology. And that is so motivating for me. And I hope I have imparted some of that passion to you. And that when you're in the lab and that mice refuses to do what you want it to do, just remember you're in it for the long haul. And that eventually good things do come from all this work that we're doing. So a couple of QR codes, publications. Metabolon has over 3,000 publications. You can go in, that's a link to the place where you can choose by disease area. You can just, you can look by matrix of interest and look at sort of different things that may be appropriate to your line of work. The resources is similar, but it's more like ebooks, presentations that are online, so that you can get, just again, a little bit wider set of information where you can get additional case studies and information that would be relevant to you in your research. So thank you very much. I'm now going to let Annie Green come and share.